Yeah, so this is my LinkedIn barcode. You can scan and add me. I'm very active on LinkedIn. I write post daily. Actually, some of what I will talk about, I already have written some of the LinkedIn posts, and you can read more about details. Okay, so well, if I ask you what the biggest issue in AI or generative AI today, I think many of you probably would agree that hallucination is probably the biggest issue. A lot of time you ask LLM, it'll give you some false answer, false results with very also repetitive tone. And people have worked on it for a long time. However, hallucination is really kind of inherent to LLM by design. Yeah, because LLM, it generates a sequence of words or tokens with probabilities. And then with some probabilities, hallucination will certainly occur. Of course, there are ways to reduce it. And but one issue with hallucination is when we ask common knowledge, usually there's no hallucination because LLM, they really know that area very well. But when we want to ask question about some subject we're not very familiar with, and usually that's where LLM also are not very familiar with, and then that's actually when they tend to hallucinate more. So basically, when we need LLM to give us answers, but then they start to hallucinate. And also we know that LLM, they store patterns or correlations. They really don't store facts. So there's no inherent way that LLM kind of do the fact checking for us. So hallucination is bad, but what about I tell you, the information used to train AI models can be wrong as well. Well, you may think, well, AI models were trained using billions of documents, and of course, they can be noise. But however, in biomedical science, it's actually a bit different because we know that for a long time, we have this so-called reproducibility crisis. For some reason, yeah, we still don't yet complete understand. Of course, everybody has their speculations, and people have found there are multiple large-scale studies that at least more than half of the published research, even in the top journals, Nature Science, they cannot be reproduced. And this is so-called reproducibility crisis at NIH now is really take on this issue is one of the top priorities of NIH. And of course, this is bad, but now we have AI, right? And then people have found out that AI actually is being used to generate papers. It's okay if you use that to write your draft, write introduction, and you proofread it. However, people have started using AI to write the whole papers, sometimes without doing any experiments. And it turns out that AI was used to just do some fake experiment, generate fake conclusions. And there are studies that showing that a lot of papers were generated by AI full of hallucinations. And then we're actually using AI to generate fake papers, and that will pollute our literature. And then this polluted literature will then be used to train the next generation of AIs, right? And then with that polluted literature, especially think about in some general domain, of course, there's a lot of noise. And then, however, in science, if people are motivated to publish and then they just use AI to publish, then we'll actually may have an even more serious issue of polluted literature. And then that worse they will generate fake paper, then this is definitely a terrible cycle. So but how can we address that? So I think the next few years, one of the major issues is we really need to generate trustable or trusted knowledge for both human researchers and also for AI models and applications. And our solution right now for hallucination, there's a general approach. And there, in fact, this is a common used approach called retrieval augmented generation. And in there also called RAG. In RAG, you don't just use LM alone, but instead you use additional data source. So when you have a question asked LM, and then this system will query data and then return some relevant information, and then this LLM will use that to generate a question with this relevant information. And then I give you better answers. In fact, this is being used very commonly in many applications. So you need setup. The data is actually very important. And a lot of times, people basically use a bunch of documents with embeddings, basically embed documents using word vectors. But that actually sacrifice a lot of explicit knowledge in the documents. And our solution to that problem is knowledge graph. So basically, knowledge graph, they are the graphs and with nodes and edges. The nodes are concepts, for example, diseases, genes, drugs, and the edges are their relations. So we can extract those relations from literature, for example, these papers or PubMed papers, and we can extract a specific relation with probabilities. So the edges can have a probability associated with them. And we can also use this relation to make inference. For example, this drug may act on a particular gene, and this gene may be a drug target for disease. Then we can infer this drug may be used to treat disease. And of course, we can make inference involving many indirect edges and make a very complicated inferences. So if we have a knowledge graph, then we can use knowledge graph in the RAG system. When people ask some question, we can query knowledge graph. And knowledge graph, because it's structured, you can retrieve explicit knowledge very efficiently. And then this retrieved knowledge can then be combined with LLM to give you much more accurate answers. Well, to construct knowledge graph, there are largely two kind of approaches. One is using the databases. The database is very convenient because most of the databases, they have structured knowledge. You can just download database, you put it into a knowledge graph. Of course, there are still issues because multiple databases, you need to do integration, harmonization, but it's relatively easy. But the other approach is to extract knowledge from text, unstructured or from literature. And that's more challenging. This is the problem we have been working on. So to construct knowledge graph from text, usually it takes two steps. The first one is called name entity recognition, or NER. So you extract concepts or entities from a literature. For example, extract the disease entities, gene entities, or drug chemical entities. And then the next step is to extract their relations. And after relation extraction, we'll have this knowledge graph. But actually, there's another step, which is also very important. After extracting entities, we need to map to official name or official ID because there are a lot of synonyms for many medical entities. Then we really need to map all these synonyms into one standard official name and ID. And here I didn't show that step, but that's actually also a very important step. So we have been working on this for actually many years since I did my postdoc at Harvard, where I worked on protein structure, modeling and simulation, and I even worked on protein structure prediction. But then I realized that problem was so difficult. So at that time, that was around 2004, there no deep learning, and so the method we used, we really don't see any hope of solving that problem anytime soon. And so before I finished my postdoc, I thought, okay, maybe I should diversify. And then we started working on some research in network biology. But to work on research in network biology, we need to have a network information, like gene regulatory information, right? But usually you get this kind of information from databases. But when we extract such information from database, we find out the database missed a lot of information people already published. Then how are we going to address that? So there's no other way. And we said, okay, then we just have to develop methods to extract such kind of information. So this is our first paper published in 2009. We extract protein interaction information from literature. And after that, I continued this line of research and carried on that to Florida State, where I was an independent researcher. And we did a large-scale information extraction from PubMed. And then we also developed a method for automated knowledge discovery. At that time, the concept really new. In fact, there's no term knowledge graph. And we call our knowledge graph, in fact, the same thing as one we use today. We call it integrated bioanalytic network. And then we did large scale extraction of different relations from PubMed literature, and then we built system for automated knowledge discovery. Yeah, but it was too early. People don't really kind of recognize it as a nice piece of work. But we continue. So you see, this one we published in 2011. It's almost 15 years ago. And over the years, we have been participating in Biocreative Challenge. Biocreative Challenge has been around for many years, and then I think this year it was nice. Every 2 years, researchers get together and they organize some challenges, and those are blended tests. Basically, organizers will manually label some data, release part of it as trained data, and then hold the other part to test participants' models. And it's kind of a good way to evaluate the state of the art of the methods. For Biocreative Challenge, the competition mainly focused on biological information extraction. So, this is a later one. Yeah, so we participated in Biocreative Challenge for many years, and I remember around 2017, and we participated Biocreative Challenge 7. 7 or 6, maybe 6, yeah. And we still use traditional machine learning, but then we find out other participants they all started using deep learning, and then they actually beat our methods. And without a lot of efforts, with traditional machine learning, you have to do feature engineering. You have actually read a lot of code to extract features. But with the latest deep learning methods, you just give these models text directly, and then they actually give you very good performance. So since that Biocreative Challenge 6 around 2017, we realized deep learning really powerful, and we started learning deep learning. And then fast forward to this is 2021, and there's another challenge called Late Core LLM Challenge organized by NIH and NASA. So basically, at that time, we already have a larger model. Google developed a BERT-based model around 2017, 2018, and then the field already started building fine-tuned models using BERT and variants of BERT models. And many information extracting tasks have really achieved much better performance. And then NIH realized that and then want to kind of evaluate the field, see how much the field have progressed in terms of constructing knowledge graph. Around that time, knowledge graph also kind of become an important topic because really I believe it's the best way to organize our knowledge in structured form, so we can really perform a lot of automated computations on this structured knowledge. So this competition consists of 2 steps. Basically, the kind of 2 steps I mentioned early on. The first step is name entity recognition, extracting entities, and second step is extracting relations. And for this challenge, they even put up a 100,000 cash prize. And the organizers from NIH, they random sample 500 PubMed abstracts, and they manually labeled all these abstracts. They labeled the terms like disease, genes, and mutations, chemical compound in these abstract. They also manually labeled a bunch of relations, association, positive correlation, etc. And it gave participants 400 abstracts, train their model, and then held 100 abstracts as test data. So we participated in the Bitcoin LB challenge, and then at that time, I was still at FSU. Our team is called Zhang Lab at FSU, and we win this challenge. And this is kind of the aha moment for me. I realized with the model we trained, we actually achieved a very good performance and close to human expert level. And then I realized, wow, a few years back, and we all know that deep learning has kind of solved the image recognition problem. Right? And then a lot of application. Now we can use our iPhone is face recognition. I know, once we actually solved NLP, natural language processing, and then there are also a lot of applications will be really kind of developed and then applied, and many people benefit. So I think, well, I probably should kind of leave academia to really develop something with higher impact. So yeah, I took a leave of absence and started working on my company full time. And in 2023, so I took leave in 2022, and actually this summer I formally quit FSU. So we at that time we have a next Biocreator challenge in 2023, that's Biocreator Challenge 8. And there's a track called end-to-end knowledge graph construction. Yeah, it's very similar to Litecoin NLP challenge. Yeah, basically the organizers get part of that data and then also enrich it with additional PubMed abstract, but it's more challenging because it's end-to-end. The participants were given PubMed abstracts, and then you have to do everything to construct knowledge graph. And then there are multiple steps. Every step, of course, the model will have to make some errors, and then cumulatively, the results not very high. And for this challenge, our team is called Insilicom, and we also win this challenge. You can see that we actually achieved performance much better than all the other teams. And since LLM, most of the team, I believe every team actually use some form of fine-tuned LLM. Yeah, for these challenges, it's actually very challenging to really differentiate because everyone built on top of probably one of the best LLM models. And but yeah, we managed to really develop some novel approaches, and then yeah. Okay, so the organizers also evaluated the latest large language model at GPT-4. It turns out actually GPT-4 performed rather poorly for this task. And they also compared, they used PubMed BERT, also the BERT model we used. And then they used two different populator models. But when we participated in the challenge, there's only populator 2 available. So you can see like populator 2 plus PubMed BERT performance is actually around 20% with the other participant teams. But using populator 3, you can get a much better performance. But populator 3 was released after our competition. Yeah, so basically if we use populator 3, yeah, we probably can also substantially improve our performance. Then after the competition, we applied our pipeline to parse all the PubMed articles. Now there are more than 37 million. And then we also integrated data from 40 public databases. We added a couple of databases. Okay, excuse me. And recently, and as a proof of concept, we also downloaded public available high-throughput genomic data. Yeah, for example, the data from TCGA, GDC, and Recount. And then we analyzed these genomic data. So after analyzing genomic data, we can actually generate knowledge. For example, we can compare cancer tissues with normal tissues, and then compare gene expression, right? And then we can get upregulated or downregulated genes for certain cancer, and that's gene and cancer relationship, right? So we can add that into our knowledge graph. So after that, we generate a rather large knowledge graph with many antitypes with many relations, and we published the paper in Nature Machine Intelligence this year. One thing I like to add is from the Biocreator training dataset and also Litecoin NLP dataset, they don't have a directional relations. That means which one causes which. They have only the correlation. But we have been working on knowledge graph for many years. We know direction is very important. Yeah, because think about it. A change of gene causing a disease is very different from you having a disease and change the gene to cause the gene to change, right? And so we then build our own causal model and then predicted the directions, which entity actually cause the other entity to change. And then we actually generated a causal knowledge graph. It turns out it's actually very important. I will show that later. So the first thing we want to look at is, okay, so we did a large scale information extraction from literature, right? So how much new knowledge have we extracted? Because people have been using these public databases for many years. And these public databases, they claim they have manually read literature and then put the manually labeled data into their database. But they have never really kind of access how comprehensive their database, basically how much coverage they have covered literature. They just say, well, no, we tried our best, we read some papers. So we then compared these public databases, and it turns out we compare several major type of relations, for example, chemical gene, chemical gene, chemical disease. And it turns out that we actually extracted a lot more than these public databases. Yeah, you can compare the scale. And because our methods we tested using LeetCode NLP challenge, we tested with Biocreative challenge, and we know of course our extracted relation they're not 100% accurate, but majority of those are actually true. That means those public databases actually missed a lot of knowledge researchers have published in the past. So yeah, there's no wonder, those knowledge graphs built using just public databases, they don't really work well because they made so much knowledge already, published. So we also compare with some specific databases. This is comparing with several databases in precision medicine. They contain mutations and then disease information. And compare with those, also, we showed that we extracted way more knowledge than those databases. So one question we often received is, in fact, it's also relevant to what I talked about earlier, because we know there's a so-called reproducibility crisis, and then even a piece of knowledge published in a paper, that doesn't mean it's actually correct. And then it's well known, a lot of published knowledge, actually, they have contradictions, right? One piece of discovery may be contradictory to something, some knowledge people already published. So how do we kind of reconcile these contradictory ones? And here we develop a probabilistic framework. We call it a probabilistic semantic reasoning. And so we can use that to infer probability between any two entities, A and B. Either they are directly related or indirectly related. So here, let's give you an example. And as I mentioned, we actually extracted a lot of knowledge from literature. And for example, any particular relations, we can extract many papers describing the relations. And if we want to infer indirect relations, there can be many entities connecting them. And then to put all these together, we really need to develop some probabilistic-based approach to infer relation between any two entities. So I'm not getting into kind of technical details. So basically, those are, although the formula is a little bit long, but they are actually very simple. They're really statistical one-on-one ideas. However, even using this statistical one-on-one to this, it took us a while. Yeah, so because you need to have a solid probabilities. And once we run our model generating all these results, as we know that deep learning models, they are overly confident. Basically, the probability produced by the deep learning models are actually not real probabilities. Yeah, they kind of highly skew towards both ends. Either they say, well, 99% or almost 0%. That's why LLM, a lot of times they tell you something with a very authoritative tone, which is actually false. But then we realized actually we can kind of calibrate these probabilities, and then after that, we design this probabilistic framework. So next, I will talk about applications of our knowledge graph in drug repurposing. There are many applications. Think about a knowledge graph, right? We have entities, we have their relations. In fact, you can predict the relation between any two entities. And then there are a lot of actually method developed working on graph data, and they call link prediction. So you can predict links between any entities. Yeah, for example, you can predict whether a drug can be used to treat a disease, or you can predict whether a gene is relevant or associated with a disease, or you can predict whether a gene regulates another disease or another gene. Yeah, basically you can predict the relation between any entities. And then you can use knowledge graph to basically generate all kinds of hypotheses. Yeah, so that's why I believe in knowledge graph will be a very important tool in AI for science because it can be used to help people to generate hypotheses. And here, drug repurposing is just one type of hypothesis. And to generate a drug repurposing hypothesis, our idea is actually quite simple. So we can start either from a disease or from drug. If we start from disease, we'll first in the knowledge graph, think about in the knowledge graph, right? We have many disease terms, we have genes associated with diseases, and we also have chemical compound drugs connected to these genes. And then we first find out drug targets for the disease. And then we find out drugs act on these drug targets. And then we can kind of infer this relationship between drug and then disease. Of course, in the middle, there you don't have to have only one gene. It may be multiple genes and connected with multiple edges. So here, for this application, you can see that the direction we inferred early on actually very important. Yeah, because we really need to infer relation that the drug will cause disease to change, right? And then the drug has to cause this drug target to change. But it can be positive correlated. Basically, drug can activate this drug target, and the drug target repress the disease, and you have therapeutic relations, right? The direction has to go from drug to disease. Or the drug can inhibit target, and the target may actually kind of promote the disease. And also you have the same therapeutic relation, right? Yeah, so in both cases, we need directionality. Otherwise, yeah, we'll generate a lot of false positives. Another thing I would like to mention is you can generate a hypothesis. In fact, there are many knowledge graph people have developed. There are many drug repurposing methods have been developed, and then every model can generate some hypothesis. But we have tried many of those, and then I think very few actually works. And one problem is these methods were not rigorously validated, right? But how can we really kind of rigorously validate? The best way is we just predict something and we wait, right? So for example, I predict something today, and I say, this is new, nobody has discovered, and then I publish. Then we wait for a few years. Of course, people won't be able to publish my paper, right? Because nobody want to wait for a few years. But we can kind of set up a similar scenario by kind of pretending we can go back to an early date, right? So for example, we can go back to 2020, and we only use knowledge published before 2020 to make predictions, and then we can use knowledge published after 2020 to validate, right? This should be the approach I think everyone should use. However, most people don't use it. They just kind of mix all the knowledge, and then they randomly sample part of it. And then they use the rest to predict the part they took away. Then there's an issue of predicting something. For example, here, because biomedical knowledge, in fact, in any discipline, right, a lot of discovery was based on early discovery. For example, discovery A is used to generate discovery B. If we just mix everything, and we might actually use discovery B to predict discovery A, which doesn't make sense. In fact, this kind of setup, not using cutoff time, that setup will cause a huge information leak issue. So what we did is we call it time-aware approach for validating our automatically generated hypothesis. So here we can have multiple cutoff. And for example, we can have a time cutoff at in 2020 or 2019, 2018. So every time we generate hypothesis using the knowledge published before that time, and then we validate our prediction using knowledge published after. So we tested our methods on COVID-19. And because in the pandemic era, COVID-19 has been studied a lot, and then we can compare with on other studies on drug purposing for COVID-19. So for this, we, using our time-aware approach, we went back all the way to very beginning of the pandemic. So we did 4 rounds of drug purposing in March 2020, April 2020, May 2020, June 2020. Basically, those are the very early days. And in March 2020, there were not a lot of papers published on COVID-19. So only 4 drug targets published. And for these 4 drug targets, we repurposed more than 600 drugs. And although only 4 drug targets, but these are actually well-studied genes. There are many drugs that can be repurposed. And in April, there were actually 8 targets published, and then we repurposed 800 drugs. And then we take these 800 drugs. We predicted using knowledge published before April 2020. And then we followed our prediction every month. We check whether our predictions were actually tested in clinical trials or later on published in PubMed, basically mentioning that drug can have some kind of therapeutic effect for COVID-19. So here, although we call it validated, doesn't mean the drug we repurposed turned out to be effective COVID-19, because we know that for COVID-19, people tried a huge number of drugs, but very few actually works. But because our exercise is generating hypothesis, right? If our hypothesis, a drug we repurpose, later on we'll test it on clinical trials. That means, excuse me, that's probably a good hypothesis, right? Because people actually want to test it. And similarly for published in PubMed. So yeah, so we tested our hypothesis, and then for every month after April 2020, and then until all the way to May 2023. Excuse me. So we have close to 300 repurposed drugs were later validated. So which is more than 30%. However, I want to point out that those hypotheses we generated, which were not validated by either clinical trial or PubMed, doesn't mean those are wrong. Maybe they're still true. We just have never tested those yet. This is already, I think, better than state of art at the time when we publish our work. One example is on genistein repurposed to COVID-19. And to our surprise, we actually identified many genes connecting genistein with COVID-19. And then for many of these genes, and we later on develop a web interface so we can display this graph. We can also click an edge, and then all the paper will show describing these relations together with the sentences we extracted from these papers. So we can quickly read about any specific relations. And in fact, yeah, since our study, we have tested for many cases repurposed drugs. And in this case, we actually clicked many of these edits, read these papers. We actually found out the evidence is overwhelming. So in fact, in 2022, there's a paper published hypothesizing that genistein can be used as potential drug to treat COVID-19. You know, during the pandemic, even hypothesis papers can be published because everybody want to help. And then, yeah, who knows, maybe they can actually, yeah, generate a use for drug. So that kind of validated our approach for COVID-19. We later on did drug purposing for cystic fibrosis in collaboration with Cystic Fibrosis Foundation. And so here we went back all the way to before 1990. Every year we do a drug repurposing. And here you can see there's a peak because around 1990, it was actually discovered cystic fibrosis was caused by a single mutation. And basically, the mechanism of the disease become known. And then we have a clear drug target. Once you have the drug target, then we can repurpose drugs. And we actually repurpose a lot. And those color, the darker bars, that means those repurposed drug later on was reported as potential new treatment for cystic fibrosis. And overall, there are between 10 to 20% were later on published as potential new treatment. And again, those are not published. That means they're actually wrong. Maybe people have never tested. So we don't really know the exact accuracy. So here, these blue lines are the average time. For example, here we have around 70 were later on validated, and this time, like 15 years, is on average how many years our prediction actually ahead of actually published time. Yeah, so then we for each year, right? For all those predictions were later on published, we know how many years we actually predicted ahead of time, and then we can calculate the average time. And then of course this trend has to be going down because in the later year, our prediction just don't have enough time to be validated, right? Yeah, for example, in the 2024, we made a prediction, but we don't have time to check whether our prediction is true or not. So therefore, this time has to be kind of going down. But if and then so this don't really reflect the reality or how much earlier we can predict, right? But if we look at around here, then actually our predictions ahead of actual published time around 15 years. That's actually a long time. And here we look at all the drugs that people publish that can be used as potential treatment for cystic fibrosis. And it turns out that 84% of those we could actually predict before they were published. That's actually a pretty large percentage. And we also did drug target identification for lung cancer. The idea is actually similar. So here instead of finding a new drug, we find the new genes that regulate a known drug target. So therefore, that gene can be considered a new drug target. And then we did the drug target identification similarly every year for lung cancer for also many years starting from 1988. And you can see that also our identified new drug target, there's more than 30% were later validated. And then the average time is also, I guess 15 years is reasonable. Yeah, so basically we can identify new targets 15 years earlier than it was actually published. And then this figure, we look at all the published drug target and see whether these published drug target can be predicted early on. And it turns out that 40% of those were actually can be predicted just using our algorithm before they were actually published. And then we look at some examples. So in fact, in drug repurposing, if you know this field, any example like this, people actually make a big deal. They say, wow, we predicted something many years ahead of, they were actually reported. But for us, we have hundreds of such cases. And here I just I select 3 for cystic fibrosis. And for example, here, tamoxifen was predicted 27 years before it was first reported. And we actually have literature evidence that you can read them. And so first, we use a paper published in 1990. In that paper, it mentioned tamoxifen can regulate the gene called VIP. And then in 1989, there's another paper published stating a relation between VIP and CF. And then we just piece these 2 papers together, and then we can propose that tamoxifen can be used as a potential treatment for CF. And in fact, in 2018, the paper published kind of proposing this indeed tamoxifen may be used to treat cystic fibrosis. So 27 years before it was first reported. And then copper. So here, copper is not just metal, it's copper ion. And we predict that 32 years before it was first reported. And then here, using two papers, one published in 1988, talk about copper regulating trypsinogen, and then another paper published in 1985, talk about trypsinogen's relation with CF. And then in 2021, the paper published proposing whether copper can be used to treat cystic fibrosis. And the third example is thalidomide. We predicted the 25 years before, and then we'll just skip this. And this is an example I published recently on LinkedIn because actually I have a friend, and then he received an email from somebody he didn't know, but it was a mother whose daughter had Wilson disease, and she was kind of asking for help because their current treatment were not very effective. So we did this repurposing for Wilson disease. And first, we just ran our Euro algorithm, and we identified a bunch of drugs. And those drugs, some were actually mentioned in literature that could be helpful for Wilson disease symptoms. But later on, we did some more in-depth research and then found out that actually this cellostazol can be repurposed and with a very plausible mechanism for treating Wilson disease. Yeah, I will not get into the details because I wrote a LinkedIn post. You can read more about it. I think it's really an interesting case and demonstrating the power of knowledge graphs for finding a new potential treatment, especially for rare diseases, because rare disease people, these large pharmaceutical companies, they don't have enough incentive to develop drugs for them, and then those patients really need effective treatments. So we recently we developed some tools using the knowledge graph. For example, this iPulse we just released recently, and we are extracting the latest discovery both from PubMed and also from conference abstracts, especially in major oncology conferences, for example, AACR, ASCO, ESMO, and we just published results from ESMO 2025 recently. Yeah, this is just a screenshot for the iPulse tool. Then so here, we have 3 different conferences this year. And then we focused mostly on drug discovery related knowledge. For example, we extract clinical trials, we extract drug targets, we extract drug protein relations and drug disease relations. And all of these, you can read about the details. And so usually, because if you look at the abstract, there are 7,000 abstract in AACR. It's almost impossible for any individual to read all of these. But we extract a structured knowledge. You can search. You can search clinical trials for any specific disease, for any specific drug, and you can get a table and then with all the results. This is another competition we participated this spring. It's called the BioASQ or BioAsk. So this competition has been around also for many years, and this year is the 13th year. Yeah, this competition has many different tracks, and the one we participated is for biomedical question answering, and our team participated in the phase A. And then we have 2 tasks. One is document retrieval, basically retrieving relevant document. This is really important task because if you cannot retrieve relevant document, you cannot really answer questions accurately. And the second task is extracting snippets, basically in the document, which part of the document can really be used to answer that question. And there are 4 different batches, and then our team managed to win first place for both problems for all these 4 batches. And then using this method we developed for question answering, then we can actually apply that to this iPulse. So instead of 6 here, you still need to go to the tables, but with chatbot. You can just use natural language. You can just ask question, and then yeah, we can get answers using the information we extracted. And I mentioned a lot of published discovery that actually wrong, and there are many papers generated by AI. They're being submitted to even well-known journals and published. Yeah, so we are actually developing this to verify published literature and also maybe used by these journals to verify these submitted papers. But because we scan all the PubMed knowledge are in our knowledge graph. So any newly submitted paper, we can check. Right? We can build knowledge graph for that piece of paper, and we can check with existing knowledge. And if there are any contradictions, we can flag them out. And this is especially useful for AI generated papers because AI generated paper will have a lot of hallucinations. Then we can flag them out, and then we can ask the author to justify. And if authors basically just use AI to read paper, they probably just give up. And then this is a very effective way to just filter out all these AI generated papers, right? Of course, that's just one application. For other people, you can just submit any paper to the system and then check how reliable that paper, even already published, how consistent that discovery with the rest of biomedical literature. Yeah, but this one we actually haven't really released it yet. We are still working on that. And we even developed a game we call the HypoQuest. Yeah, this game we can actually we release it internally, then we plan to release it soon. We can have a college student, even high school student play it. Yeah, think about it. We have a huge knowledge graph, and if somebody come with a question, for example, you know your friend may have a rare disease. Well, you may want to find out a new treatment for that, and then you can use our knowledge graph, and then you kind of search in this. We call it navigate in this knowledge graph, right? You can go from one node to another node, right? You can just keep searching and then generating null hypothesis. Yeah, that's what we call it, a HypoQuest. And we're making it kind of a game-like. We are gamify this so that people can compete, that they can achieve goals, badges, and then we will organize events, competitions, and then we plan to work with foundations to organize events so that these foundations, the disease foundations, may actually sponsor events for the community to compete with. So finally, we would like to build a knowledge graph we call it foundation knowledge graph to serve as a trusted layer for future AI models and applications. Yeah, think about the future AI models and applications. If they solely rely on LLMs, which are trained using polluted literature, then we are really kind of generating all kinds of hallucinations. But with a foundation knowledge graph, then we can use trusted knowledge to build trustworthy AI models, and then that will help us generate trusted knowledge. So basically, we turn this vicious circle into virtuous circle. All right, so I think that's pretty much what I want to talk about. Any questions? Thank you.